What is the name for enzymes that increase the rate of a chemical reaction without being consumed in the process? A. Catalysts. B. Proteins. C. Substrates. D. Inhibitors. The correct answer is A. Enzymes are biological catalysts that speed up chemical reactions without being used up in the process. Which of the following is a property of enzymes? A. They are consumed in the reaction. B. They only work at specific pH levels. C. They increase the activation energy of a reaction. D. They are highly specific in their actions. The correct answer is D. Enzymes are highly specific in their actions, typically catalyzing only one type of reaction. What is the IUB classification number for an enzyme that hydrolyzes peptide bonds in proteins? A. 3.4.21.1. B. 2.1.1.1. C. 1.1.1.1. D. 4.6.1.1. The correct answer is C. Enzymes that hydrolyze peptide bonds in proteins are classified under 3.4.21. Which type of enzyme inhibition happens when a molecule binds to the enzyme, but not at the active site? This binding changes the enzyme's shape, making it harder for the substrate to bind. A. Competitive inhibition. B. Non-competitive inhibition. C. Uncompetitive inhibition. D. Mixed inhibition. The correct answer is B. Non-competitive inhibition involves binding to an allosteric site on the enzyme. What is a common therapeutic application of enzymes in breaking down blood clots that cause heart attacks and strokes? A. Digestive enzymes. B. Proteolytic enzymes. C. Oxidoreductases. D. Isomerases. The correct answer is B. Proteolytic enzymes break down blood clots. They're beneficial in treating heart attacks and strokes. What term describes enzymes with different amino acid sequences but the same reaction? A. Active enzymes. B. Competitive enzymes. C. Isoenzymes. D. Substrate-specific enzymes. The correct answer is C. Isoenzymes are enzymes that catalyze the same reaction, but have different amino acid sequences. Which of the following is a key role of coenzymes in enzyme-catalyzed reactions? A. Provide structural support to the enzyme. B. Act as substrates for the enzyme. C. Transfer functional groups to or from the enzyme. D. Stabilize the enzyme-substrate complex. The correct answer is C. Coenzymes assist enzymes by transferring functional groups to or from the enzyme during reactions. What is the term for the process where an enzyme changes shape when a regulator molecule binds, making it more or less active? A. Allosteric regulation. B. Competitive regulation. C. Feedback regulation. D. Inhibitory regulation. The correct answer is A. Allosteric regulation involves a change in enzyme shape due to binding of a regulator molecule. Which enzyme class is primarily responsible for transferring phosphate groups between molecules in biological systems? A. Oxidoreductases. B. Transferases. C. Lyases. D. Phosphorylases. The correct answer is B. Transferases are enzymes that primarily transfer functional groups between compounds, such as phosphate groups. What is the term for the maximum rate at which an enzyme can convert substrate to product when its active sites are fully occupied? A. Km. B. Vmax. C. Cut. D. Turnover number. The correct answer is B. Vmax is the maximum rate of reaction catalyzed by an enzyme when saturated with substrate. Which of the following enzymes is classified under IUB number 1.14.x.x and is responsible for transferring an electron between compounds? A. Oxidoreductases. B. Transferases. C. Hydrolyzes. D. Lyases. The correct answer is A. Oxidoreductases are enzymes that catalyze oxidation-reduction reactions where electrons are transferred between compounds. Which type of enzyme inhibition happens when a molecule binds to the enzyme outside the active site, changing the enzyme's shape and blocking substrate binding? A. Competitive inhibition. B. Non-competitive inhibition. C. Uncompetitive inhibition. D. Mixed inhibition. The correct answer is B. Non-competitive inhibition occurs when an inhibitor binds to an allosteric site on the enzyme, causing a conformational change that reduces substrate binding. What is the term for enzymes that can run their reaction backward, depending on substrate and product levels? A. Unidirectional enzymes. B. Bidirectional enzymes. C. Irreversible enzymes. D. Regulated enzymes. The correct answer is B. Bidirectional enzymes can catalyze both the forward and reverse reactions, based on the concentrations of substrates and products. Which isoenzyme is commonly used in cardiac diagnostics to detect myocardial damage? A. CKMB. B. ALP. C. AST. D. LDH. The correct answer is A. Creatine kinase MB, or CKMB, is an isoenzyme of creatine kinase mostly found in the heart muscle. Elevated CKMB levels in the blood indicate myocardial damage. What role do coenzymes typically play in enzyme-catalyzed reactions? A. They act as structural proteins for the enzyme. B. They provide additional substrate for the enzyme. C. They act as catalytic centers. D. They assist in enzymatic reactions by carrying chemical groups between substrates and the enzyme. The correct answer is D. Coenzymes act as carriers of specific chemical groups between the enzyme and the substrate, facilitating enzymatic reactions. Which enzyme class mainly adds or removes groups from a compound's double bond? A. Lyases. B. Isomerases. C. Ligases. D. Hydrolyzes. The correct answer is A. Lyases are enzymes that catalyze the addition or removal of groups to or from a double bond in a compound, leading to bond cleavage or formation. In enzyme kinetics, what term describes how efficiently an enzyme converts substrate into product? A. Turnover rate. B. Km value. C. Specific activity. D. Catalytic efficiency. The correct answer is D. Catalytic efficiency is a measure of how effectively an enzyme converts substrate into product, indicating the enzyme's efficiency in catalyzing a reaction. What is the term for enzymes that work best at extreme pH levels, like low pH for acidic enzymes and high pH for basic ones? A. Neuroenzymes. B. Allosteric enzymes. C. Extremozymes. D. Regulatory enzymes. The correct answer is C. Extremozymes are enzymes that work best in extreme conditions. These conditions include extreme pH, temperature, or salinity. Extremozymes are often found in organisms that live in harsh environments, like places with extremely high or low pH levels. What is the primary function of coenzymes in enzyme-catalyzed reactions? A. Act as structural elements of the enzyme. B. Provide energy for the catalysis. C. Bind to the substrate directly. D. Carry chemical groups between enzymes and their substrates. The correct answer is D. Coenzymes play a crucial role in carrying chemical groups between enzymes and their substrates. Enzyme kinetics typically measures the efficiency of an enzyme through which parameter? A. Activation energy. B. Allosteric regulation. C. Michaelis constant, Km. D. Catalytic efficiency, Kcat over Km. The correct answer is D. Catalytic efficiency, Kcat over Km, measures how efficiently an enzyme converts substrate to product. Which type of enzyme inhibition occurs when a molecule binds to the enzyme at a site other than the active site, altering its shape and reducing its activity? A. Competitive inhibition. B. Non-competitive inhibition. C. Uncompetitive inhibition. D. Mixed inhibition. The correct answer is B. Non-competitive inhibition involves binding to an allosteric site, changing the enzyme's shape, and reducing its activity. Which enzyme class mainly catalyzes adding or removing phosphate groups from molecules in biology? A. Oxidoreductases. B. Transferases. C. Hydrolyzes. D. Lyases. The correct answer is B. Transferases are the enzymes primarily involved in transferring phosphate groups. In enzyme regulation, what term describes an enzyme changing shape after binding a regulatory molecule, thus altering its activity? A. Competitive inhibition. B. Cooperativity. C. Allosteric regulation. D. Feedback inhibition. The correct answer is C. Allosteric regulation describes the alteration in enzyme activity due to binding of a regulatory molecule. What is a common therapeutic application of enzymes that break down proteins into smaller peptides and amino acids in medical treatments? A. Chelation therapy. B. Enzyme replacement therapy. C. Prodrug activation. D. Antibody-directed enzyme prodrug therapy. The correct answer is B. Enzyme replacement therapy involves using enzymes to break down proteins into smaller peptides and amino acids. Which term describes enzymes most active at extreme pH levels, like very acidic or basic conditions? A. Neutral enzymes. B. Isoenzymes. C. Extremozymes. D. Orthologous enzymes. The correct answer is C. Extremozymes have optimal activity at extreme pH levels, whether acidic or basic. Which of the following is a key characteristic of isoenzymes? A. They exhibit differences in amino acid sequences but catalyze the same reaction. B. They have identical sequences and catalytic properties. C. They are only expressed in specific tissues. D. They do not require any coenzymes for activity. The correct answer is A. Isoenzymes, or isozymes, are enzymes that catalyze the same reaction but have different amino acid sequences. Often, they are expressed in different tissues or under different conditions. What is the term for enzymes needing a coenzyme or cofactor to work properly? A. Allosteric enzymes. B. Metalloenzymes. C. Apoproteins. D. Holoenzymes. The correct answer is D. Holoenzymes are enzymes with two parts. A protein part called an apoprotein, and a non-protein part, either a coenzyme or a cofactor. Both parts are needed for the enzyme to work. Which enzyme class is mainly responsible for adding or removing phosphate groups from molecules in biology? A. Ligases. B. Isomerases. C. Kinases. D. Hydrolyzes. The correct answer is C. Kinases are enzymes that catalyze the transfer of phosphate groups between molecules. They play a crucial role in signal transduction pathways. Which term describes enzymes working best at extreme, very acidic or basic, pH levels? A. Allosteric enzymes. B. Isozymes. C. Metalloenzymes. D. Extremozymes. The correct answer is D. Extremozymes are enzymes that show optimal activity at extreme pH levels, including highly acidic or basic environments.